Good morning, Misty. How are you?
Hi, great, Sarah.
Welcome to Merck TV. We're doing a special interview today with Associate Professor Dr. Misty Jenkins. We're going to learn all about her research in cancer and immunology. So welcome, Misty.
Thanks, it's lovely to be chatting to you today.
So Misty and I have known each other for about 10 years. I'm going to kick this interview off with a very big thank you, because as a young researcher coming through the ranks, you and I were in completely different fields of immunology, but from the moment I met you, you had my back, both pumping me up to have the confidence to speak at conferences and talk about my poster sessions, to talk about my poster, but also to have my back in a way that woman to woman, not a lot of people are able to do. So I want to say thank you, and you have been an inspiration to me, and I hold a very big core memory of you and have been following your career ever since. So thank you, Misty.
Oh, that's absolutely my pleasure to know you and to love you and support you, because I think you're amazing.
Misty.
Right back at you.
Oh, beautiful. So more than 10 years we've known each other. We met at an immunology conference. I think it was a lawn infection and immunity conference. And like I said, we're in different fields of immunology. You were more focused on lymphocytes, more focused on T cells,
Yes.
and really in the human disease space, and really, cancer. And, tell us how you got to be a cancer researcher.
Well, I actually did you're right, I have a long-standing interest in cytotoxic T lymphocytes, and, trained across the road here at Melbourne Uni with Peter Doherty and Steve Turner, looking at T cell responses to viruses, actually, to flu. And so I was always interested in how the T cells form their immune response and how they play different roles of recognising different antigens to work as an orchestra. And then it was only in more recent years started collaborating with mentors and colleagues at Peter Mac, and then took a real interest in cancer, because actually the way the T cells kill the cell, whether it's a virus-infected cell or a cancer cell, the mechanisms are conserved. They are the same. So I had studied that process for many years, and then at Peter Mac really grew my interest in cancer immunotherapy, because that was at a time when immunotherapy was really starting to make waves, and it was just fascinating.
So how did you get to WeHi from Peter Mac?
So I did my second postdoc at Peter Mac and spent 5 glorious years there with Joe Trapani, who's a superstar. And then when I started my lab at WeHi, of course, with everyone starting your lab, you have to, work on something that is different. You have to find your own niche, and you have to carve out your own area of research that's different to your mentors, and your supervisors. And knowing at the time that Peter Mac weren't really interested in, didn't currently have any work in brain cancer, but also this was about the time in my life when I met my now dear friend, Ruth Mitchell, who is a neurosurgeon, and she's now a paediatric neurosurgeon. And she was doing her PhD here at WeHi with Tony Burgess. And she said to me, I'm sick of telling my patients they're going to die, and someone needs to do something about that. We're throwing all these brain tumours into the bin and no one's researching brain cancer, and you really need to have a look at it. And at the time, what I thought was a fairly serendipitous conversation between two friends, I look back now and actually think that it was such a turning point in my career. She really lobbied me hard. And then I started reading, and I knew nothing about brain cancer. I knew nothing about brain cancer. I was a card-carrying immunologist, Sarah, yourself. And so but when I did start reading, actually it became pretty obvious this was a pretty perfect marriage, scientifically speaking, because unlike a lot of other drugs and antibodies that don't get into the brain and cross the blood-brain barrier, T cells do, and in fact our brains have quite an active immune response and system. So that was it. I was hook, line and sinker, and was absolutely resolute to start my lab using immunotherapy to treat brain tumours.
So tell us more about the Brain Cancer Centre. What have you been able to achieve here in the Cancer Research Institute?
The establishment of the Brain Cancer Centre has been a bit of a game changer in team science. For team science. It's been a big win for team science, because we have challenged this model of one lab and you're a bay of 6, and one lab and you're a bay of 6. We're working genuinely collaboratively. So every little, piece of brain tumour tissue that comes directly out of the operating theatre, we now have systems in place that they come straight into our labs. We have committees that decide the priority of tissue across across the whole organisation, so that we can all analyse that brain tumour tissue and different aspects of it. I look at it from an immunology perspective and mapping the surfaceome, the proteomic surfaceome of the tumours, so we can generate new targets for therapy. But I work very closely with the lab doing metabolomics on the same sample, or spatial transcriptomics to understand the tumour microenvironment. And then we also work very closely with the organoid team and generating models of these tumours, and also with the drug development team in terms of screening. So only by doing that can we actually then link all of that data at the end and actually learn a lot more about the tumour biology and responses to it.
So what kind of translational information are you able to provide back to the patient and back to the clinicians once you've received that biopsy sample?
Yeah, absolutely. This is where I think this is what the Brain Cancer Centre through BPOP and our clinicians do very well. So we've been the first in Australia and the world actually to initiate a phase 0 trial for adults with glioblastoma. And what that means is that the brain tissue is analysed and taken before standard of care, and then the patient might go on to have surgery or radiation and chemotherapy, and then we can look at the responses later and also take tumour. So you can look at a longitudinal, you can look at how the tumour changes in response to therapy and drugs, and then you can also intervene early and look at the effects of those interventions. So it's been a real game changer.
Is that the future? Is a personalised approach like this the future?
In my opinion, yes. I'm very biased, but I do honestly think that we've moved beyond these blunt tools, these blunt instruments of radiation and chemo will always have their place, but really being able to understand the tumour at a molecular sort of omic and proteomic level, and then to personalise the subsequent treatments, and whether they are drugs, antibodies and checkpoints and immunotherapies in combination with other small molecules, that is absolutely the future.
And how long do you think it will be until you and the lab are out of the job, so to speak, using your words, for cancer research in the brain?
We'd like to do ourselves out of a job, wouldn't we? Unfortunately, brain cancer is a really tricky disease, and I think if you look at other indications like breast cancer, for example, that had huge investments 30 years ago that absolutely transformed the prognosis now of breast cancer, that's taken 30 years. And so I think we're probably looking at a similar timeline in the brain cancer space, but it is going to take it's going to take more horses in the race. It's going to take a large investment of money, and of course that involves people. People are the major cost, really. And working collaboratively in that multidisciplinary way to really solve the problem. The problem is as well, of course, that unlike other solid tumours where you can take good margins and say in the case remove a whole breast, for example, or organs, you can't do that in the brain. And the tricky thing about some of these poor prognosis brain tumours like glioblastoma is it's like throwing a handful of black sand onto a white beach and picking out the grains of black sand. It's very hard to surgically remove all of the tumour. And so surgery is great because it gives patients time in a lot of cases, but we're going to need to go for cure, particularly in the paediatric space. Brain cancer kills more children than any other disease, full stop, which I think is pretty horrifying. More kids get leukemia, more kids die of brain cancer. And so whilst it's rare, it's terminal in a lot of cases, and you can't talk about 5-year survival rates with a 5-year-old. We have to be going for cure. It's an incredible field of research that you're in, Misty. Besides cancer, is there anything else that piques your interest around immunology and the kinds of pathways that you've understood for your whole research career? Well, I think I've sort of focused mainly on the kind of killing soldier immune cell type in the CD8 T cell, but of course there's a lot of immune cell types, as you will know, that are immunosuppressive, and brain cancer is no different in that sense. So there are a lot of immune cells that we don't understand the role of in the brain that I'm really interested to unpack.
Yep.
So we're using some really cool technology actually to use cover slips implanted into living brains of mice, and then using 2-photon microscopy to penetrate deep into the tissue and image longitudinally over time a lot of these immune responses in the brain.
Wow.
And what I'm really interested to understand is the role that the microglia in the brain are playing, the interaction of those cell types with the CD4 and the CD8, you know, CAR T cells, if you like, and how we can then overcome the immunosuppressive tumour microenvironment inside these brain tumours. So I think there's still so much we don't know, and because the brain is a really difficult place to work, it's been challenging, I think, to look, but I think we're now starting to have the technology, like these implanted in cranial windows in combination with 2-photon, to really start to unpack some of these questions.
Wow.
And of course, the single cell omics, you know, has been spatial omics has been amazing. So, you know, single cell spatial transcriptomics, you know, which is now all the rage over the last 12 months, is I think really helping elucidate the roles of these different cell types and how they're talking to each other. And then, of course, with the view of, okay, once we understand the biology of that, we can then intervene and design interventions to block, suppress, and activate.
So, Misty, I want to talk about Deadly Science. Merck partnered with Deadly Science in 2021. They're our charity partner of choice. We're teaming up with them to develop the first STEM education kit for remote Indigenous communities in Australia. We have our first trip out to Robinson River this year to implement this kit. I know Indigenous STEM education is something very close to your heart, and that you're also partnered with Deadly Science through WeHi. So tell us all about it.
Well, first of all, I'm thrilled to hear that Merck has partnered with Deadly Science. I think that's pretty deadly. I first met Corey a few years ago through CSIRO and was just so inspired. My mum's Gundi Jamara, so I'm from Western District, Victoria, my mob, and so working in Indigenous health and education's always been just a part of who I am. And was thrilled, you know, to get WeHi on board to partner with Deadly Science. We've now hired a program coordinator, Joshua Waters, who's brilliant, and we're working with Deadly Science to bring, you know, that Indigenous community perspective into WeHi, and also, you know, to showcase our scientists out in remote and rural communities, and in fact all Indigenous communities. You know, I think a lot of people forget that, you know, most Indigenous people live in cities as well. So so all of Australia, it's a national and very much a national project, and I'm just really privileged to be a part of it.
This is an amazing work that you're leading in collaboration with your peers.
Definitely.
But so impressive, Misty. Thanks for sharing that story with us, and where the future of research is, and how you got to where you are. It's very, very impressive. Thanks for your time, Misty.
Thank you so much, Sarah, for having me.
You're welcome.